So the discovery of the CRISPR-Cas9 system has revolutionized the field of gene therapy and led to the rapid development of CRISPR-Cas9 based therapies to treat inherent diseases. Most Cas9 autologs are derived from common bacterial pathogens to which more than 80% of healthy people have antibody and also T-cell responses. And previous studies could show that there's a pre-existing Cas9 immune response towards Cas9 homologs of Streptococcus pyogenes and Staphylococcus aureus. However, immunity towards therapeutic gene vectors or gene-modifying cargos can negate the success of treatment and can possess significant safety issues. As Cas9 editing is advancing towards the clinic, it becomes necessary to address the inherent immunogenicity in order to guide clinical treatment decisions. The existence of Cas9 antibodies is likely not relevant, as antibodies rather mediate the opsonization and phagocytosis of pathogens and cannot penetrate through the cell membrane. So antibodies targeting an intracellular protein may likely not lead to the killing of the cell expressing that protein. So the killing will rather be mediated by cellular immunity or more particular T-cell mediated responses. So most gene therapeutic approaches aim at delivering the Cas9 protein directly into the target cell or expressing the Cas9 nuclease in the target cell, which leads to protein degradation processes and the presentation of Cas9 on the target cell, which can be recognized by T cells. However, this scenario can raise safety concerns for in vivo gene editing, while ex vivo gene editing may be less risky, because we could also show that in gene edited T cells, the Cas9 concentration decreased in a time frame from a week, so after the gene editing. And also our group could show that the Cas9 directed immunity does not only include Cas9 specific effector T cells, but also regulatory T cells. And these regulatory T cells, they could suppress the proliferation and cytokine production of Cas9 specific effector T cells. Consequently, these Cas9 specific regulatory T cells can be a tool to suppress or counteract the effector T cell responses in vivo, especially in those patients who have an unbalanced Cas9 specific regulatory T cell to effector T cell ratio. And also the unbalanced ratio of effector T cells and regulatory T cells should be monitored carefully in clinical trials in order to gain information whether the Cas9 induced immunity may be tolerogenic or rather dangerous. Because an unbalanced ratio with high frequencies of CD8 T cells can lead to risk stratification for future trials and may also exclude high-risk patients from receiving a CRISPR-Cas9 based therapy. Though that's why we aim for the generation of a GMP-compliant protocol or GMP-compliant T-cell product, the enrichment and expansion of regulatory T cells to be used in the clinic as an adaptive T-cell therapy approach in combination with CRISPR-Cas9-based therapies to provide or ensure the success of these therapies. Because antigen-specific regulatory T cells can be a targeted and living immunosuppressant to induce immune tolerance and to support the success of gene therapies.